Good morning, this is me Nafisa and today we'll be learning about phagocytosis. What is phagocytosis? Or what is the meaning of phagocytosis? Phagocytosis is made up of three words. Phagen in Greek meaning to eat, cytos meaning cell, and osis is a process. Phagocytosis is the process by which a cell engulfs a pathogen, or we can say that a cell chews up the pathogen, or a cell eats the pathogen, or we can say that the cell phagocytoses the pathogen. What is the role of phagocytosis? Or what is the main function of phagocytosis? Phagocytosis process is used by the phagocytes to kill the pathogens that have successfully passed through the first line of defense. Phagocytosis forms a part of the second line of defense. Immune cells that perform phagocytosis are known as phagocytes. Examples of phagocytes are neutrophils, macrophages, dendritic cells. There are a number of distinct steps involved in phagocytosis. Step 1 is the activation of the phagocyte. First, the phagocytes should be activated. It should be alerted that there is an invading pathogen that needs to be cleaned up. The result is that phagocytes gain the ability to leave the capillaries and enter the tissues towards the site of infection. This is known as chemotaxis. So what is chemotaxis? It is made up of two words, chemo meaning chemical and taxi meaning movement. So chemotaxis means the movement of phagocytes towards chemical signals such as pathogen or complement proteins or cytokines. So step 2 is the chemotaxis of phagocytes. Activation also enables phagocytes to produce PRR on their plasma membrane. What is full form of PRR? Pattern recognition receptors. These PRRs will recognize and bind or attach to the pathogen's PAMPs. PAMP full form is pathogen associated molecular patterns. Now please note, PAMPs are not found in human cells. PAMPs are components of pathogens alone. So step 3 is recognition of invading pathogen and attachment of the phagocyte to the pathogen. Once the phagocyte recognizes the pathogen and following attachment, the phagocyte will surround their cell membrane around the pathogen and ingests the pathogen, or I can say absorbs the pathogen, or internalizes the pathogen by forming a sac known as phagosome. So step 4 is ingestion of the pathogen by the phagocyte and formation of phagosome. This phagosome fuses with lysosome to form phagolysosome. So step 5 is formation of phagolysosome. Now as we know, lysosomes is full of digestive enzymes and lysozymes and antimicrobial substances. The resulting phagolysosome has an acidic environment and variety of enzymes which together result in killing of the pathogen. So step 6 is killing of the pathogen. After the pathogen is killed and digestion of the engulfed pathogen has taken place, only indigestible or non-digestible waste remains in the sac, which is known as residual body. So step 7 is formation of residual body. Finally, the residual body then moves to the cell boundary and is removed out of the cell as waste. This process is known as exocytosis. So step 8 is elimination or exocytosis.